Hello, and welcome to a new Lupus podcast. We record these podcasts for you every month, so you will always be able to keep up to date on the latest literature and expert opinions. I am Professor Vibeke Strand. I'm an adjunct clinical professor in the Division of Immunology Rheumatology at Stanford University School of Medicine in Palo Alto. Today, I'm joined by Professor Chi-Chu Mok, the Chief of the Division of Rheumatology, Department of Medicine, Chuan Moon Hospital, Hong Kong. He's a consultant physician and an honorary teaching professor of both the University of Hong Kong and the Chinese University of Hong Kong. Today, we're going to review two key publications from October covered on the Lupus Forum. So I will start with a phase 3 trial of telitacicept for systemic lupus erythematosus. So this was a phase 3 trial. These were patients with active systemic lupus, and the typical inclusion criteria, a SLE score of 8 or at least a SLE score of 6 with positive anti-dsDNA and complement anti levels, low complement levels. And essentially, we were looking at telitacicept 160 milligrams versus placebo. And the endpoint was at 52 weeks. And the primary endpoint was the SRI-4, although they have altered the SRI-4 definition because they use a SLE score of 4 or more improvement, but then they use no BILAG A or 1B as opposed to A or 2Bs, which is considered to be a more meaningful BILAG worsening. And so perhaps they may have caught a few patients who would have worsened otherwise. And then, of course, there was no change in the PGA. The secondary endpoints included multiple endpoints as we are used to seeing. And basically what we saw was that if you look over on the right-hand side, out to 52 weeks, telitacicept was doing much better than those patients on placebo. They had, of course, been on background medication, typically mycophenolate mofetil and glucocorticoids. They were asked to maintain the glucocorticoids stable after 24 weeks so they could change them before then. And then they were asked to try to taper at 48 weeks up to 52 weeks. Now you see with the figure that the response was 67% in the patients 112 who were still on telitacicept and not treatment failures versus 32.7% in the placebo patients, 35 and 35, sorry. And you see that the percentage difference was highly statistically significant. So we know that more patients who received telitacicept than those receiving placebo had a response by this modified SRI-4 criteria. And this started as early as week 4, and the difference was sustained through to week 52. Although there was no signal of increased serious adverse events or really a difference in the safety profile other than telitacicept was associated with more upper respiratory infections, decreases in immunoglobulin levels, and increased injection site reactions. Dr. Mok, what do you have to say?
Yes, I think you remember we have similar products, similar RCTs with similar trials performed in Europe and America about another product called telitacicept. And that product was prematurely terminated because of infection. And in this study, it also causes hypogammaglobulinemia and increase in infection rate. So what do you think? Why the two products have different results? Apart from modifying the SRI-4, what can cause the difference in the result? And if you look at the SRI-4 response in the BLISS renal studies, it seems that the percentage is much higher than the BLISS study. It is even better. The response is better than the BLISS study.
Yes, well, I agree with you. Not only is this response better than what we saw in the atacicept trials, But it was also better than in the other BAFF/APRIL inhibitor that now has moved on. But telitacicept now is turning out to be approvable in IgA nephropathy. What do you think of that?
I don't know. I think there's some commercial reasons why the drug is not further developed. I think if they can perform it because LPC study, they may show some good results of this product blocking both the BAFF and also the APRIL. And actually, telitacicept was approved in mainland China after the successful phase 2. And in the past few years, and this product was very frequently used in SLE patients in mainland China. It's not available in Hong Kong, but it was commonly used. And there was many and real-world study published from the mainland China colleagues on the efficacy, and it was reported to be better than belimumab, subcutaneous belimumab. So I wonder whether in the future, if we have such a product approved for system lupus, and recently we have the obinutuzumab, the more potent anti-CD20. So I wonder if, for severe manifestation, which induce the patient with anti-CD40 and then followed by telitacicept or telitacicept maintenance. So what do you think?
Yes. And it's interesting too. I mean, telitacicept's been approved in China for several years now, has it not?
Yes.
And is it widely used in lupus?
Yes, because it is produced by the Chinese companies and it's very, very frequently used in mainland China. Or usually it's for refractory lupus, for the patient with suboptimal response with the conventional and immunoprotective drugs.
And then we've also seen a positive study in Sjogren's, so we should see more with telitacicept. And perhaps finally, a North American and European trial.
Yeah. So maybe in the interest of time, we have to move on to the second study we are going to discuss. The second one is a RCT, another RCT, phase 3 RCT, and a placebo-controlled RCT on the efficacy of secukinumab in anti-lupus nephritis by Professor Zhao, Beijing, China. And we know that secukinumab is a monoclonal against IL-17A. And this drug has been approved in psoriatic arthritis, skin arthritis, and spondyloarthritis. And there's evidence that the Th17 helper T cells and the double negative T cells, which mainly produce IL-7, is increased in the lupus mice and also in the human lupus nephritis samples. And in both the mouse model and also in the human model, in human patients, and the IL-17 level correlate with disease activity and severity of the lupus nephritis. And there are several case reports that secukinumab is effective in refractory lupus nephritis. And therefore, this RCT looks at whether there's efficacy of secukinumab over placebo in patients with active class 3 or 5 lupus nephritis. Recruitment is adult patient still positive, and include patient with a standalone lupus nephritis proven by biopsy, and randomized the patient to receive 300 milligrams and subcutaneous secukinumab versus placebo. And the dosage is high. To me, it's high because the licensed dosage for arthritis is usually 150 milligrams. They use a high dose, 300 milligrams weekly for 4 weeks and then monthly. And the core study, the kind outcome is efficacy at week 52. And the criteria were similar to the pre-LN study, but the protein threshold is 0.5 instead of 0.7. And instead of 10% drop in eGFR, they use 15% drop and also more than 16 ml per minute of the eGFR. And the second endpoints include the change in urine protein, a partial renal response, time to a complete response, a partial response, and the fatigue score, the quality of life, as well as the adverse events. And the study, both the core study, extension study was terminated because a scheduled interim efficacy analysis at week 52 show futility. That means the CRR in the secukinumab group was only 24%, but in the placebo is even lower than the placebo group, and the CRR rate is 6.3. So regarding the adverse event, and there's no difference between the two groups, except some special interest. Like in the psoriatic study, we observed an increase in candidiasis. And in the core study and also in the extension study, there was a numerical increase in oral candidiasis as well as pneumonia. And there's also increase in herpes zoster in the extension study. So overall, this is a negative trial. And if this is a post-concept, IL-17 blockade alone is not beneficial in the treatment of lupus nephritis.
So are you surprised by these data?
No, because I think it's very complicated disease, lupus, and it's driven by multiple cytokines. You know, the property of cytokines is very pleiotropic, and redundancy is a characteristic. So if we just block one single cytokine, it may not be successful. And there's some other examples, like we blocked the 23, the secukinumab was just published earlier this year. This is again negative study. And we also know that use secukinumab, which block IL-12 and 23, also not successful. So blocking one cytokine may not be a good idea. And we need to block multiple cytokines, for example, by the CHEK inhibitors, and it may be more promising result with blockade.
So I did notice in the inclusion criteria, they did not have a required SLEDI score at baseline. Now, I know obviously this is lupus nephritis, but for instance, the US FDA always asks for a measure of lupus disease activity in a nephritis trial. Do you think that made a difference?
Yes, because this just released and I haven't gone into the details of the supplementary data and supplementary protocol, etc. And there's no mentioning of the SLEDI requirement on study entry. And perhaps they have the data, but they will publish in another publication. But I'm sure for this sort of studies, they should have the SLEDI data.
Yeah. Well, I was still surprised because I thought that the earlier trials gave us some idea, or the earlier studies gave us some idea secukinumab would be helpful. But I think that part of this was the trial design, and part of it was the secukinumab is not sufficiently helpful in lupus nephritis. Is that your conclusion?
Yes. And also, in the discussion, the authors criticized a very high placebo response in the placebo group. Indeed, this is true because the criteria they use, the CRR, is slightly more stringent than the primary endpoint PERR in the Lupus LN study. Now, despite this, the placebo response is 36%, which is even higher than the PERR of 32% in the Lupus LN study. So I don't know how to explain this, why the placebo response will be so high. Even the patient was treated with the conventional lupus cyclophosphamide and MPA mycophenolate together with a post-steroid oral steroid regimen. And this is one of the reasons why this study failed.
Yes. Well, that's all time we have time for today. Thank you for joining me, Professor Chi-Chu Mak. It was great to hear your thoughts on the latest developments in lupus management.
So, and thank you for listening. You can find the slide decks for these papers at the lupusforum.com. The Lupus Forum is free to access, and all our content is free to download. If you are not already, make sure to register for updates on the Lupus Forum. You will receive email updates when new content is made available. You can also follow us at Lupus Forum, all one word, on Twitter and LinkedIn. Thanks again and see you next time.